that's a relatively small market. So they've been trying to move up and trying to encourage people to say, well, actually treat earlier. Don't treat when they're at 8, treat when they're at 10. Don't treat when they're at 10. Iron deficient, and this is the message they're now saying, is iron deficiency is a chronic ongoing condition. You want to manage it. Now in Switzerland, that message has resonated with the doctors from quite a long time ago. So if a patient goes in and even looks tired, they're likely to be strapped to a machine and given IV iron. And there's, it's a home market for them. Of course, the more that V4 push that story and are changing the paradigm to managing iron deficiency, by far and away the best way of doing that is with an oral therapy in a chronic setting.
Are we in Switzerland yet?
We are.
The drug is approved in Switzerland, and we have a commercial partner, and they're going through the pricing and reimbursement at the moment.
Yeah.
So, and actually, the regulators have been very accommodating, actually, with therapy. We've had a very constructive negotiation and discussion with the regulators in Switzerland. They clearly want, the impression being they clearly want something else in the market to change the paradigm.
I've got a more technical question now. Somebody drew to my attention, in fact it was last week, I don't know if he's here, no it's not. It was a paper out of the Netherlands, and it referred to how iron can affect the microbiome.
Yes.
Now, so the question is does that give the IV formulation an advantage over therapy? And is the problem with the microbiome getting altered the same for ordinary iron tablets as it is for therapy, or is one superior over the other?
Great question. I always like to cover food in these stories. No, I mean, I think it's a big issue, and it's a particularly big issue in the IBD population where, the change in the microbiome, there's a lot of debate about whether that's causing a lot of the exacerbations and things are going on. The fundamental reason why you get a change in the microbiome is free iron in the gut. It's the free iron that's causing the issues around the bacteria and changing the dynamic of the bacteria from a survival point of view, so it changes the balance. There is a paper that's going to be presented at BSG of work done with...
so BSG is the British Society of Gastroenterology meeting.
Yes. That's in June. So I can't talk about the paper in itself and the outcomes of it, because it was a piece of research that was done by someone else. But the principle of why the microbiome changes would say, because ferrocru doesn't leave free iron in the gut, and because the amount of iron anyway is much lower, you would not expect to see the same changes in the microbiome. And I think that's down to the mode of action. It's a perfectly reasonable hypothesis. IV iron shouldn't cause the changes in the microbiome, as you say, because it's not going in through the gut. But ferrocru could well look different in that study compared to oral irons because of the lack of IV oral iron. So I'd keep a lookout for that.
And is it a clinical study, or is it in animals, or is it?
It's a combination. So they've done a mouse model, which is the most controlled model. Obviously, humans are awkward because humans have a tendency of going down the chip shop when they're hungry or out of beer and eating things that they would normally eat. So they can change their diet. They've done a mouse model, which is a very well recognised model where they can completely control the feed. So that's part of it. And they've also done some work in humans as well. So they've taken sort of stool samples from patients before and afterwards and tried to balance that. So they'll have a bit of that.
Yeah. So is this coming out before the FDA? I mean, it's a great thing to have in your back pocket if you get questions.
It's in June at the BSG, which I think is the 18th to 20th of June.
Okay.
I think Glasgow.
Look out for that. Did you sponsor it?
We supported that piece of work, but it was done independently.
It won't play into the FDA's deliberations, though. We provided them with a dossier as of the 28th of September, and that's the dossier that we provided them with. So, for example, the head-to-head data, we've not submitted that. We could have done, but there would have been a clock stop, which probably would have stopped the clock for about 6 months, and we really didn't think that was worth it.
We didn't need that data.
We didn't need it. Yeah. And it'll eventually go into the label, it'll go into the clinical section of the data sheet in the US.
You'll have some of the safety data from the updates that we've done, so that data is in there, but not yet.
Yeah, the microbiome question, the authors of that Netherlands paper seem to think it's more of a developing world problem than a developed world, so...
Yeah, no, think the microbiome's a very interesting area, and one that is getting a lot more interest.
I think it goes towards the physiological adaptation of ferrocrude, and the design of it such that it works with the body and not necessarily against it.
I've got loads more questions. I expect Andrew has as well. But
is there anybody else with any questions?
Carl, are you expecting an advisory committee?
No. Look, FDA, you can never say never, but no. If they're going to, they better tell us soon.
Yeah, well, we went through there's a new process that's not long run about where you basically sit them down and talk through your file at the beginning of the process, so you explain where everything is, and the sort of application orientation. So that, if you get that done well, then they understand where everything is, and it makes the review process a lot easier. So that's the deal, but we went through that with them, and I think it has helped, actually.
Well, the obvious question to me is, surely you're vulnerable to a takeover, particularly from the US partner, because if they look at potential sales in the US and the sort of royalties you got on the other deal, it'd be cheaper to take you over than to sign a deal.
You know, the only way I can really answer that is that my job with the cards we dealt with, my job is to return the maximum value I can to our shareholders. So we have a number of defensive strategies to try and, if somebody wants to offer enough money to the shareholders and the shareholders say yes, then that's going to happen. There's not really a lot any of us can do about that. If enough money is, because anybody who's a shareholder, if people in here are shareholders and they say yes to such an offer, then you're making that decision, not me. My job is to make sure that we get maximum value if that's the case, and we have a range of strategies to do that.
You acknowledge that's a risk.
I think it's a potential outcome. I'm not sure it's a risk, but I think it's a potential outcome.
Wrong word, yeah.
People originally just offered cash to you. They didn't want to take stock in lieu and leave it for cash.
We didn't give them the option. No, we wanted the cash. The company needed some cash anyway. It needed a cash injection at the time, and we didn't want to take too much. But if we could have taken 20, maybe 25 million up front, but actually we didn't need 20, 25 million. We didn't need, so to speak, the 11 million. We needed less than that. But if we'd have taken 20, 25 million, we probably would have had royalties in the high teens because of the effect it has on a discounted cash flow model and the NPV that creates. Because obviously cash here and now has a big value to it. Whereas we could see where this was going, we could see the value of this product. Mike's here. When Mike and I first met, I had a light bulb moment in the first sort of 5 minutes. I had a background in cardiopulmonary medicine and orphan diseases, and those patients have a lot of anemia, so I knew the complexities. And when I saw some of Mike's data, it was like, I knew the problem and I could see the solution. So consequently, we understood the value of the asset, and having also extended the IP out the way we have, to give away those royalties, I think would have been a mistake.
I think one of the graphs showed it takes 12 weeks roughly to get to where you want to be. If you then stop treatment, how long does it take to go back to baseline, if you like? And if you want to maintain the optimal level, are you still on the same dose, or do you reduce the dose, or just do it once every 2 days rather than?
So, firstly, as a physiological process, so the 12 weeks gets you up to where we get to. Actually, some patients will take longer to get to their natural hemoglobin. So it depends on the patient group. So for you and I, if we stopped taking iron and we didn't have any underlying disease, the red cell turnover is 168 days, 180 days. So by the time you're turning over red cells, you lose a little bit of iron from that. It would take quite a long time. Obviously, if you've got an active disease state where you're bleeding into your gut a bit more, then it will happen quicker. So it does depend. I mean, as Mike said, we know that patients who are having IV, you give them it, they're back again. Some patients are back again in 6 to 8 weeks. Some patients take longer. But it depends on the individual.
So to expect the same thing to happen, but maybe longer.
It may take longer. At different disease states, different severity of disease states, it will take longer, but you will come back down again.
So if you want to maintain them at the target level,
Yep.
do you carry on giving them the same?
Well, you see, this is the benefit of a physiological control.
Yeah.
Is if you give something that is well tolerated, say you're giving 60 milligrams of elemental iron, if the patient needs 30 milligrams or 20 milligrams of elemental iron, they'll absorb 20 milligrams of elemental iron. If they need 10, because they're actually stable, and they'll take up 10, and the rest of it will go through. If they need 30 or more, the more they need, the more it'll take up.
And they don't know how much they're taking up.
And it changes over time. So you might have a situation where someone's actually quiescent in the disease, they're not needing much, they're not getting side effects from the tablets, so you leave them on it. But then they have a little bleed, but rather than actually that having an effect on their hemoglobin, you top them up. So it's a completely self-balancing. It's an
active absorption.
It's an active absorption that is controlled by what the body needs. It's physiological.
And Mark's point then about the patient having a little bleed is really important about the iron stores, because the body will, as soon as it has a bleed and its blood levels go down, the body will upregulate production of red cells, and therefore it will demand more from the iron stores. And if you don't have the stores because the patient hasn't been taking the therapy to make sure the stores are there, then you have a problem. So
it's stored in the liver.
Yeah.
Can I just reinforce that? Many years ago, we did a very silly study. Sir Richard Thompson, who Carl met, myself and Bob Hyder was a co-inventor on some of their works, and we took up to 5.75 grams of ferrochrome. We didn't hyperabsorb. The only thing it was highly calaminic and made you feel sick.
Yeah.
If we'd have taken that amount in ferrous sulfate, we'd be dead. And in the United States, they have poisonous unions for children, and there's 6,000 deaths per year on existing ferrous products. So I think there is a very big, strong safety argument to get ferrochrome through to these people, so that the hospitals and the poisons units don't have to deal with children who take their mother's pills.
Yeah. That's a great point. And the safety side of things, obviously one of the challenges of the ferrous salts is iron, it's actually quite a small molecule. When it's in there, free iron, free ferrous, it can be absorbed, or it can get through any minor abrasions in the gut. It can be absorbed straight through the wall, so you can get this challenge. And DMT is not particularly a selective molecule, a selective receptor. So the thing about ferric maltol is it's a huge molecule as ferric maltol. It's enormous. So it doesn't get absorbed in that same way. So yes, the design allows for very good absorption, but prevents unwanted absorption. And in a situation where your body, as I say, can't eliminate iron, it's really important that it only takes up what it needs.
So the IP challenge, TIVA from memory, isn't it? So could you expand on that a little? I don't know the background or what they're challenging.
So I'll try and keep it fairly untechnical, probably because I'm not qualified to be technical about it. So we have a whole range of patents that protect the product, from clinical use to manufacturing through to composition. There's a period in Europe where once the European Patent Office have so-called granted a patent, there's an appeal period or an objection period, I should say. So TIVA have effectively a trawling department, and they look for approved products with any IP that's still going through. This is our understanding, that they're still going through. Perhaps some IP is going through a review process, and they look for bargaining chips, I guess. So we had a patent that was we have a core patent which is, upon approval in Europe, had supplementary protection certification added to it, so we got an extra 5 years of coverage. And we also have some patents to protect around workarounds around that core patent. And that was the first objection. It was to one of those patents. So we rejected it, but the European Patent Office decided it should go to a hearing, which, why wouldn't they? They've got absolutely nothing to lose, because it's the two companies that have to do everything. So we went to a hearing, and we had a list of 7 alternative positions to take. Obviously 1 through 7 being the least change. And within 5 minutes of the Patent Office's of the hearing, they'd accepted our first change. and threw everything else out. And that one change was instead of, in layman's terms, instead of it saying you take ingredients A, B, C, D, E, and F, and you throw them together, it said take ingredient A, add it to ingredient B, then add it to ingredient C, etc. So that a person with the appropriate skills could follow the methodology, and then it's all been finalised. So that's how the patent is now referenced on the EPO website. And excuse me. Of course, Teva can still object to that. They have a period of time. So anytime in the next 6 months, they could object to that. We'll just have to wait and see. The second patent is something that we filed in 2014, which is we demonstrated that Therapeutics exists in different crystalline forms, and they have objected to that on a number of levels, but it's all, at the moment, it's all theoretical objections. We have refuted all of those. We'll have to wait to see what the EPO decides in the sense of do they think there's something to have a discussion about. And if they do, both parties will eventually get called to a hearing. And then, as would have been the case with the manufacturing patent, if it gets found in our favour, then Teva can appeal. If it gets found in their favour, then we have the right to appeal, and the process will go on. Or there'll be an ability to reach a middle ground. It's a strange one though. So the composition patent clearly extends our IP. So the main manufacturing patent has protection with the SPC, and eventually it'll get some pediatric extension as well, out till well into 2029. And the composition patent added that to 2035. So the drug is protected no matter what until 2029. And will Teva even be around in 2030? If they were to win that patent, if it was to be completely extinguished, and they had an absolute 100% victory, then what happens in 20? It's a bit strange. We're trying to work out if there was a market now, which there clearly isn't in therapy. It's not got 250 million sales right now. If there was a market now, then you can understand why they were trying to challenge it. But even if they succeeded right now, they still would be locked out of the market until 2029.
So it won't affect your sales to 2029, whatever.
Yeah. It is a bizarre one.
Can I add a slight technical detail, because that particular patent, the several work around that, I was very much ignored. I'm the inventor. Teva objected on the grounds of obviousness, saying that the way of which the Ferrocru was manufactured, starting off from a salt base, is distinct from the one which is currently used. And they cited 3 publications. Those 3 publications are in fact, on analysis, not relevant to the methods that we claimed in that patent. And the agent looked at that situation, and that's the basis on which they lost their case. Originally, the opposition division were favouring Teva. But once we looked at it in detail, it wasn't. And in fact, the 3 publications didn't produce Ferrocru half the time, they produced ferric oxide. And so they couldn't produce a pharmaceutical quality product by the 3 methods that they cited in the public domain. And that's why they basically, I think, have lost their case. And if they get to come back, they'll have, in my view, have got a lot of problems in trying to actually persuade the opposition division that in fact that patent should be.
Well, actually, the point being really that even though it was a workaround patent, when we look at, we know that we have the most efficient, the least cost manufacturing process anyway, and that patent will still hold.
I think you said even if all patents were overturned, you'd still have exclusivity out to 2029. Is that strictly true? If they were prepared to repeat clinical studies, they could get an early?
No. So what I said was, if all of the tenets of the composition patent were overturned, which takes out to 2035, that does not affect any of their IP at all. So the existing IP that has the supplementary protection certificate, which was granted upon approval of the drug in Europe, goes out till currently 2028, but with the paediatric extension, when we finish that study, actually when we finish our commitment to the PIP, we'll take it out to 2029.
So they can't just repeat your whole program and and get another license?
No.
Oh, okay.
If they were able to knock the manufacturing patent over, which we don't believe they could, I mean, really, and they'd have to have evidence. They can't just appeal it. That path has been in existence for a long time now, so they would have to actually show fundamental evidence, nothing theoretical, that they have knocked it over. Then, and that's a huge amount of work. And of course, the drug is also protected by data exclusivity post the approval in Europe Act, so that goes out until 2026, February the 28th, 2026.
Can I ask about food hasn't run?
It has.
It has.
But don't worry, you can ask one more question. I'm just inching up slowly.
I think last time we spoke I wasn't a shareholder, I have bought something, so I've been doing my homework. Your pipeline, you told us about PT20, perhaps only renewables I'll hear more about that, but there was two others in there, there was PT30 and PT40. Now, I think one was you were going to collate it with sucrose, in which case my question is why? And was PT30, was that an IV formulation? And
So, yeah.
if so, why?
Okay, so both of them are. The commercial rationale for it, I'll explain. Mark can maybe give some more clinical, medical perspectives. So as good as Ferrocru is, it's never going to have 100% of the market. So if you can get a little past the market, why not? And all the existing IV ions have a label that requires possible administration because of the risk of anaphylaxis or hypersensitivity, etc. So we thought we'd done some work with some scientists in Cambridge around PT20, the phosphate binder, and when we got into discussions with them, they'd also done some basic research around the generation of targets that would be hyposensitive or hypoallergenic intravenous ions. So that's the idea behind PT30, and we sponsored a 3-year research program at Cambridge for this. The first year they took all the existing IV ions to identify how they cause their problems, because there are different ways of causing the problems. Year 2 they then tried to theoretically design molecules that would not cause those problems, and then year 3 they created some of those molecules in a test tube. And at that point we put a temporary halt to PT30 because we needed the capital. We felt it was better to spend the money on Ferrocru, to be honest.
Okay.
But it sits there as it went. PT40 also came out of that program, so at the end of the first year we sat down with them to see where the team had got to, and they were talking about all of the work they'd done to identify how the existing products cause their problems. And they talked about a drug called sucrose, or Benefer is the trade name. And this is a drug that we've looked at, maybe using it as a comparator for Ferrocru in the approval process back in 2010 when we were first reading, 2011 when we were first talking to regulators. And the regulator said to us, you can't use that because if it came for an approval now, and this went off patent in 2008, this drug, if it came to approval now, we wouldn't approve it because we've got no idea how they make it and what's in there. And the inter-batch quality. And then this research scientist said to us, as a throwaway comment, because he was much more interested in PT30, obviously in the program, and we've worked out how to make Benefer, and all of our ears pricked up. Okay, so this is a $200 million a year market at the moment, which the product has been generic since 2008, but there's been no genericization of the opportunity. So we have sponsored some work to see if we can scale up those. So at that point in time, we said to them, stop, make it. So they made it in a test tube, and then we have sponsored some work, and that work is ongoing at the moment to see if we can scale up, because it's fundamentally different to go from a few milligrams in a test tube to being able to make grams, kilograms of it under a sustained quality program, etc. So if that was to work, we've already had discussions with the FDA about how we would then get that approved, and that would go through what's known as the ANDA process, which would give us some priority as well, so we get exclusivity. A number of companies have tried it and have all failed. I think it's because they don't actually understand the science behind it, and we've got a bunch of guys in Cambridge who frankly, are the world's leading nanomolecular specialists.
Okay.
So we'll see how it